So picking up where we left off, we have two true breeding flowers, big P, and little p. We create the F1 generation, which will all be big P, and because it has a big P, the flowers will all be purple. But when we go to the F2 generation, we have independent uh, or law of segregation, so only one parent only contributes one. So here we can have big P, we could have little p, or we can have two versions of big P, little p. 3 out of the 4 of these are going to appear purple. The last one will appear white. So a couple of useful terms to come up with here. One is when you have two identical alleles, such as big P, or little p, we call that homozygous, right? Um, Conversely, if you have two different alleles, so when you have big P and a little p, we call that heterozygous. And unlike the homozygotes, heterozygotes are not true breeding, right? So when you are heterozygote, you can contribute two possible versions to your offspring, and it'll be up to chance of which version they get. Now, because of the different effects of dominance and recessive alleles, an organism's traits do not always reveal its genetic composition. So we need to make a distinction between what the organism looks like, its physical appearance, which we refer to as the phenotype, and what its actual genetic makeup is, which we call the genotype. So phenotype, what it appears to be, genotype, what it is genetically. So when we look at these 4 possible versions that we can get in the F2 generation, we can get big P, that is homozygote. Its phenotype will be purple, and its genotype will be big P, homozygous. Both of these two copies here are phenotypically going to be purple, so you would not be able to tell it apart from this one, but they are both, in fact, heterozygous. The final one here, its phenotype is white, and its genotype is going to be little p. In this case, you can actually figure out what its genotype is based upon its phenotype. And this is because this allele is recessive, and we'll get to that here in a moment. So when you look at the ratios here, phenotypically you will have a 3-to-1 ratio, and this is what Mendel was able to pick up on. Now, looking at it in retrospect, we see that genetically there's actually a 1-to-2-to-1 ratio. So the ratios are going to be different whether or not you're talking about phenotype versus genotype. When you are trying to figure out whether or not the purple flowers are in fact heterozygous or homozygous, the easiest way to accomplish this is to do what we call a test cross. And this is where instead of breeding them with themselves, you simply breed it back with this recessive white parental strain, which will give us a better understanding. This is something that we call a test cross. So an individual with a dominant phenotype could be homozygous or heterozygote. What we can do is breed that mystery individual with the homozygote recessive, and that is what we call trust cross. If the offspring display the recessive phenotype, then the parent must be heterozygote. So an example of this is we have this purple flower. We don't know if it's big P, or big P. So if we cross it with a little p, there are only two possibilities. If it is homozygote, then all of the plants will be purple, just like when we do an F1 generation. If it is heterozygote, so big P, little p, you will end up with this scenario here where we have half the offspring being white and half the offspring being purple. Because you can see here the recessive genotype, little p, will always contribute a little p. So half the time the purple flower will give you purple, and half the times you will get the recessive allele. So this is an easy way of determining whether or not you have big P, little p, or whatnot. Now, when we start looking at multiple traits being passed together, we need to start looking at different issues. So here we have a yellow pea that is round. So it is a true breeding, so it will have big Y, meaning that is yellow. Little y would mean green. Big R means that it's round. Little r means that it's going to be wrinkled. So here we take a yellow round pea, cross it with a plant that produces green wrinkled peas, and then we are going to produce an F1 generation. This will always be yellow round peas because these are the dominant alleles. But there are two possibilities that could fall out here. So if these genes somehow travel together, you would end up with this scenario. You take the F2 generation, and when you give a big Y, you would always give a big R. When you gave a little y, you would always give a little r. So this is the idea of dependent assortment. And over here, same thing is true. So these are the only possibilities you'd get. You'd still end up with a 3-to-1 ratio. Now, it just so happens that this is not the scenario that turns out most of the time. Occasionally, it does turn out this way, particularly when two genes are very close together on the same chromosome. Most of the time, what you get is this thing called independent assortment. So in this scenario, the Y's segregate separately from the R's. So you could produce a sperm that had big Y and big R, a big Y, a little r, a big Y or a little y and a big R. Sorry, that somehow got moved. Or you could have a little r, little y, little r. If you line that up across the top and you have the same possible combinations for the eggs across the Y, now you can start filling in the genotypes and fill in the you can start filling in the genotypes and then predict the phenotype based upon that. So this would get big Y, big Y, big R, and it would be yellow and round. This one here would get big Y, little r, and little r, so it'll be yellow, but it'll be wrinkled. This one right here would receive little y and little y, so it'll be green, but it will get a big R, so it'll be round. This one over here will get little y, little r, so it'll be green and wrinkled. So here, phenotypically, we would get a 9 to 3 to 3 to 1 ratio. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 1, 2, 3, and 1. So when Mendel did this, he happened to get this 9 to 3 to 3 to 1 ratio. And it just so happened that all of the true breeding varieties that Mendel picked happen to be ones that do in fact sort independently. It was very lucky for him because if he had picked a slightly different variety, he may have ended up in a scenario where most of the time he was getting independent assortment, but sometimes he was getting something else. And that would have been really, really confusing mathematically to figure out. So Mendel was very lucky and he picked things that did sort independently. So he was able to prove that this is the general process, is that most of the time these individual characters assort independently. All right, so utilizing a dihybrid cross, Mendel developed this law of independent assortment. It states that each pair of alleles segregate independently from another pair of alleles. So yellow will segregate independently of wrinkled, and purple flower will segregate independently of yellow or short or tall. So this law applies only to genes on different non-homologous chromosomes or those that are far apart on the same chromosome. When genes are close together on the same chromosome, it is unlikely that the recombination or the crossing over event that occurs during meiosis would actually separate those genes or separate those two different characters. So, genes located near each other on the same chromosome do, in fact, tend to be inherited together and show a dependent assortment. But most of the time, independent characteristics are independently assorted. So, looking at concept 14.1, Mendel used scientific approach to identify two laws of inheritance. So, one is the law of um, independent assortment, which we just talked about. The other is the law of segregation. So you can only contribute one version of your alleles to your offspring. So, a little review here. We have different inherited characters, which we could call genes. Those genes have individual possible traits, which can be the different alleles. So for each character, an organism inherits two of these alleles, one from each parent. If you get a difference of alleles from different parents, one will tend to be dominant and determine the organism's appearance. The other will tend to be recessive. And then we have the law of segregation. So when you produce gametes, you can only give one version. So we looked at how this plays out, both genetically and phenotypically in terms of purple and white flowers. This idea of putting things on this grid is something that we call a Punnett square. And in class, we will do some exercises where we practice producing these Punnett squares. You can see here there's a difference between phenotypic ratios and what the actual genotype ratios look like. You can see that if we need to know whether something is heterozygote or homozygote, if we do a test cross, we can easily determine that more easily than doing a bunch of self-fertilizations. And we also looked at the possibility of doing Punnett squares with more than one characteristic. When you get beyond two characteristics, a Punnett square is very difficult to write out. So typically we limit this. But we also saw that genes, for the most part, are going to sort independently.